science friends. Happy snowy NTI day. I hope you all are doing well today. I'm going to start off by saying I will be checking your notes when we get back. That is how you will get your participation grade for NTI. So make sure you watch this video through and get the notes for it to get your full grade for that when we return. So when we left off, we were talking about pedigrees. And so remember, pedigrees are used for, they're kind of the family tree to look at to track a genetic trait through a family. So today we're going to talk about different types of human traits. So one thing I want to start out with is discussing this slide. Traits are genetic. Of course, we have talked about many traits that are genetic over the course of this unit. But there's also a huge environmental component in traits. We see that a lot in identical twins. So in identical twins, there are a lot of differences. You know, one of them may develop cancer, one of them may not develop cancer. So even though their genes are identical, there are environmental factors as well. Nutrition, exercise, where you live, toxins and different things that you're exposed to. Lifestyle choices, all of those things are important. They're not inherited, but they can change your DNA in a way that you pass down, okay? So it can alter genetic or environmental decisions and nutritional factors. All of those things can actually alter your DNA in a way that you pass on to your children. So the decisions you make don't just affect you, okay? So the human genome, in the early 2000s, scientists completed the human genome, and it was a huge ordeal. And so the human genome is looking at every single gene in the human body that codes for a trait and what it codes for. And so this was an undertaking for like 10 or 20 years. Scientists worked on this to try to decipher the human genome. And so it's just all of the genetic information in human DNA. So if for example you saw a picture of a karyotype for your unborn child, okay, and you noticed there was a chromosomal disorder, maybe chromosome 15 was one of them was a little bit shorter because there was a piece of that chromosome missing. Scientists could look at what section of that chromosome is missing, and because of the Human Genome Project, they know exactly what genes have been deleted, and so they would be able to predict the type of effects that would have on your offspring before they're ever even born because of the Human Genome Project. Okay, so thousands of genes that have been fully sequenced. Okay. One gene and one trait that we're going to talk about today is the human blood group. So blood is always interesting to students. We talked about blood a little bit when we talked about multiple alleles. I think we did that on a virtual lesson once. But there are 3 alleles that code for blood group: I big A, I big B, and little I. And so it is a trait that is multiple alleles. And because of that reason, there are multiple different blood types that you can have. You can have type A, you can have type B, you can have O, or you can have AB, okay? Now, so blood group shows multiple alleles. It also shows codominance. Blood type I and blood type A are codominants. And so that means if you have AB blood, then you show the A antigens on the surface. That's a marker for A blood. And you also show the B antigens on the surface. That's a marker for B blood. Okay? Now, little I is recessive, and so there's no antigens or no markers on the cell surface for that. Okay? Now, we're going to get into antigens and markers in just a minute a little bit more. Now, people always ask, what about the positive and negative? So that's called the Rh factor. Rh, it was first discovered in rhesus monkeys, and so that's where that Rh comes from. And so that's a different gene, okay? So there's a gene that codes for blood type, and then there's a gene that codes for positive or negative. So those two genes are not really related per se, but they both code for different things about your blood. They may even be on completely different chromosomes. I'm not exactly sure on that, but they're not necessarily related to one another. So Rh positive is dominant, okay? So that means Rh negative is recessive. You have to get 2 of the recessive alleles to have negative blood, okay? So you will need to draw a chart similar to this in your notes. I think there's a place for you to draw that. I'm going to add a little bit to this chart as well. So if we're talking about O blood, I like to think of O blood as just a plain simple circle, okay? It has no antigens or markers on the surface. So antigens are things that your body recognizes. And if you give it an antigen it knows, it will accept that blood. If you give it an antigen it has never seen before, then it will not accept that blood. It would reject that blood type. We'll talk about that more in just a minute as well. Okay, so A blood is the blood cell, and it has, I'm going to make this look like triangles. It has triangle-shaped antigens on the surface. Okay, B blood has circle-shaped antigens on the surface. And AB blood, therefore, has both shaped antigens on the surface. And that's why it's considered codominant. You see the traits of B and the traits of A both expressed in that AB blood. Okay? So, me personally, my blood is type A. Okay? That means if you give me a triangle, any kind of blood with a triangle on it, I know what to do with the triangle. If you give me something with a circle on it, my body does not recognize that circle-shaped antigen. And so my body would try to reject that. So what type of blood can I take as an A person? I can take A blood and I can take O blood because O doesn't have any of those antigens on the surface. Okay? I can donate to A blood and AB blood, because A blood recognizes those triangle-shaped antigens, as does AB blood, okay? O blood is only going to be able to accept O blood. They do not recognize those triangles. They do not recognize those circles, and so they will not take that, okay? Now, O blood, though, everybody can accept O blood. The A's, the B's, the AB's, and the O's. And so for that reason, O is considered the universal donor. They can donate blood to anyone. Specifically, O negative, okay? Positive people can only get negative people can only get negative blood. Let me write this since I said it wrong. Negative can only receive negative. Positive can receive positive or negative, okay? Negative blood can only receive negative. So anybody then can receive O negative blood. So that's the universal donor, okay? B are going to be able to get from B and O and give to B and AB, very similar to the way that A could, okay? And then finally, AB blood is only going to be able to donate to other AB bloods, okay? AB cannot give to just simply A or simply B because they don't recognize that other antigen. That's considered foreign and their body would try to reject that blood, okay? However, AB can receive blood from all 4 types. So they are called the universal recipient. And specifically, AB positive is going to be more universal than AB negative. They could take any kind of blood at all. Okay? So, AB has the A and the B antigen, the circle and the triangle. B has just the B antigen. A has just the A antigen. And O blood has no antigen on the surface of the red blood cell. Okay? So that's the phenotype. Let's also look at the genotype. Okay, so the genotype for AB is I big A, I big B. The genotype for B would be I big B, or I big B little I. Okay, A is going to be similar, I big A, or I big A little I. And then O is going to have that completely recessive blood. Okay, so that is how blood group works. Now, let's talk about a few other disorders in humans. These are autosomal disorders. Now, what do we mean by autosomal disorders? That means that they are not on, not on a sex chromosome. They are on an autosome, and so that is a non-sex chromosome. So a few recessive alleles, albinism, I think you all know what that is. Cystic fibrosis, that's where they have mucus that builds up on their lungs, causes a lot of respiratory problems. Phenylketonuria, that's abbreviated PKU. Okay, they cannot break down the amino acid phenylalanine, which is, it's an amino acid that makes up proteins. That causes mental retardation. If they consume too much phenylalanine, it'll be toxic to their tissues. Achondroplasia is dwarfism. Huntington's is another one. They usually don't develop symptoms until they're like in their 40s or whatever, and their mind just starts to deteriorate. And then sickle cell disease. So sickle cell disease, normal blood cells are round. Sickle cell blood cells are kind of banana shaped like a sickle. And so they have trouble carrying oxygen to all of their blood cells. Okay, so these up here are recessive. These are dominant. And then this is a codominant allele. So a sickle cell would mean that's codominant. Some of their blood cells are normal and some of their blood cells are sickle shaped. That's how it's considered codominant. Okay. One thing that I want to point out is just because a disorder is dominant, like these, does not mean that it's dominant in the population. Okay. Achondroplasia, for example, or dwarfism, is a dominant disorder. But most of the population is not dwarfs, okay? So just because it's a dominant disorder does not mean it's dominant in the population. Another great example of that is polydactyly, which means having like more than 5 fingers and 5 toes on each hand or foot. And so that's a dominant disorder. But again, it's not dominant in the population, okay? So many of these are a very small change in DNA. Cystic fibrosis, for example, sickle cell disease. Sickle cell disease is usually a change in one base. Like we're talking about changing CUG to CCG. Something like that can cause sickle cell disease. It's more prominent in the African American population, but it can happen to anybody. But it's one little letter change, but it greatly changes the protein and causes a serious genetic disorder. Okay. All right. So let's talk about your assignment for today. You should have a paper in your binder that looks like this. It says Genealogy of the Gods. So it's going to take you through some Greek mythology, Zeus, all of those people, Apollo, and you have to come up with the genotypes down here, okay? So I'm talking I big A, little I, all right? Now, I will tell you, there is one person on here, and I don't remember off the top of my head which one it is, that you can determine their exact phenotype, but you can't determine the exact genotype, okay? So, for example, one of them, and I'm not even sure I'm going to tell you the right answer, but let's say you know that they have type B blood, okay? But you're not sure if it's I big B, or I big B little I. There is one person on here you will have to give 2 genotypes for that reason, because it's either an A blood or a B blood, but you don't know exactly which genotype it is, okay? So, this assignment would be 11 points normally because there's 11 questions, but for that one, that makes it an extra point. So it's a 12-point assignment for this part. Now, I believe on the back of yours, there is also something that goes along with the blood typing game. So, linked on the class calendar is a link to a blood typing game. I believe it's only going to give you one patient, and you will have to give it blood. You've got a patient, you'll have to draw their blood, determine what type of blood they have, and then you will have to give them blood. Okay? So, on that paper, you will have to say those things. You'll have to tell what type of what type of blood your patient had and what type of blood you gave them. Okay? Now, your assignment may have 3 different patients on it because the game used to be structured where you had 3 different patients, but I think they changed the game recently, and so you only actually need to do it for 1 patient. So if there's extra spots on your worksheet, that's okay. Just do the game for 1 patient. And again, the link for the game is on the class calendar. All right? If you have questions, let me know. If not, have a great day.